I can't fit the whole thing into my throat
Yep.
without enzymes because they help break down the stuff.
Yeah, thank you very much. Yes, Faizan, you this old or you just raise your hand? Faizan?
Yeah, I was gonna say that it depends how fast you can chew stuff,
Yeah. Okay,
like how
guys,
fast
so
you can eat.
thank you all. Thank you all. That's why enzymes are important, that yes, that act as a biological catalyst. And as we said, enzymes speed up chemical reactions that take place in the cells. Enzymes act by lowering the activation energy. Here, this is the graph, which is important to understand it. As we say, the reactants need activation energy to become products. The activation energy is lowered by presence of enzymes, okay? Activation energy without the enzyme would be higher than the activation energy with the enzymes. Enzymes are very specific. We explained this. And for this reason, part of an enzyme's name is usually derived from the reaction or the reactant it catalyzes. Enzyme action. For a chemical reaction to take place, the reactants must collide. Look at this. You know, any chemical reaction, the reactants should collide together, right? Touch each other to do the reaction. They will not react together wireless, right? They have to what? Collide with each other, right? With enough energy. And this collision should be in enough energy so that exciting bonds will be broken and new bonds will be formed. This is the chemical reaction. Again, we have two chemicals. When they collide to each other, okay, with enough energy so that would break down the bonds in these two reactants and rearrange the bonds in different way. This is the product, right? Here, if reactants do not have enough energy, if the reactants in the energy, in the reaction do not have enough energy, so what to do? No reaction will take place, right? You have here, for example, these reactants, but they do not have enough energy to collide with each other to become product. So the reaction will not occur. By the way, this reaction is the activation energy, which is needed to start the reaction. So here the enzyme work. The enzyme bind with the substrate to form enzyme and substrate complex. Like when this is the enzyme, okay, and these are the substrates. When the now enzyme and substrate, when the enzyme or the substrate bind on the active site here, when they bind on the active site, now what we call this? We called it enzyme substrate complex. Okay, it's the enzyme and the substrates are bind on its active site. Enzymes provide a site where reactants can be brought together to react, reducing the energy needed for this reaction.
Mr. Can you let Diego in?
Yeah, okay. So as you see, it does two things. Bring the reactants close to each other and reduce the energy needed for the reaction. Okay? If you keep them out like that without enzyme, they need to collide to each other. So they need to come close together and they need energy enough to break down their bonds and rearrange it again to become product. Here the enzyme provide this, okay? The reactants of enzyme the enzyme catalyzed reactions are known as substrates. We said this. The substrates, glucose and ATP, bind to active site on the enzyme. For example, the glucose and ATP, the energy form, they bind on the active site on the enzyme Hexokinase. This is the name of enzyme. May you ask, oh, this is glucose and this hexokinase, how? You just said that the energy or the enzyme name can come or derived from the name of substrate. Yeah, remember glucose is C6H12O6 and it has hexon shape, right? So that's why the enzyme called hexokinase, okay? Forming an enzyme substrate complex, okay? The fit is so precise that the active site and substrates are often compared to lock and key. As we said, this is the old theory, but today it's induced fit. This is old PowerPoint actually. This PowerPoint very old. It's supposed to be now induced fit theory compared to induced fit theory, okay? The enzyme and substrates remain bond together until the reaction is done and the substrate are converted to products. The products of the reaction are released and the enzyme, look at this, very important. The enzymes now are free. The enzymes now are free to start another process with another substrates, but of the same type, okay? Remember, enzymes are not used up in the reaction. They just provide suitable conditions. They bring the reactants close to each other, reduce the activation energy that needed to start the reaction. Okay?
Okay.
Okay. Here
Mister, I have a
you can
question about
see,
slide 3.
yes, Ali.
Can you send this PowerPoint in Classroom?
Yeah, it's already on class, Abdullah. Yes, Abdullah. Yes, Ali,
Yeah.
Rosan.
On slide 23, the slide before this.
This one.
Yes. What do you mean by hexokinase?
Hexokinase is a name of enzyme that used to react glucose with ATP. Now the glucose and ATP, they want to react together. Here, there's enzyme provide a suitable site, active site to bind these substrates, the glucose and the ATP. So these ATP and glucose react faster, right? What we call this enzyme, which is specific for glucose and ATP only. Only glucose and ATP can bind on. Got it? What we call this enzyme? Hexokinase. Anytime you see ASE, so this is the name of enzyme. Hexokinase. What else? Huh? You can say saccharase, as we just mentioned. Huh? Lactase, amylase. Do you remember amylase? Salivary amylase, amylase, ASE, end by ASE.
Yeah.
This is the name of enzyme, okay? Here, we have 1 minute, then we finish. We have the glucose and the ATP. You can see how they bind, the substrate bind to the enzyme, and they form enzyme substrate complex. The enzyme and the substrate bind it to each other, so this is enzyme substrate complex. Now, the substrates will convert into product and then release the product out. The enzyme is hexokinase is free to start with another glucose and ATP. Got it?
Yes.
Yeah, next time we will talk about factors affect on the enzymes work. Okay? Factors affect on the enzymes work. Next time, inshallah. Yalla, have a nice day. Do you have a question?
Yeah.
Yes, what?
What's the product when glucose and ATP combine?
What is the product
Yeah.
of, what you said? Product
Glucose
of?
and ATP. Glucose and ATP.
Yeah, wait, let me open it on the slide. Stop sharing and then go to this slide. Yes, here. You are talking about this one or the product or the product itself? The product?
The product itself.
Yeah, the product. Actually, here, when the ATP react with the glucose, actually the glucose need the phosphate group. The glucose need the phosphate group from the ATP. So they react together to get a phosphate group. The phosphate group, this one, will bind, the blue ball, will bind on the glucose during the reaction. Got it?
Got it.
Yeah, so that makes glucose 6-phosphate. We called it. This is the name of product. Glucose 6-phosphate. So now this one is different than the glucose. Don't say, oh, still same. How the reaction? No, glucose 6-phosphate is different than glucose only. Okay. And what about the ATP? ATP had 3 phosphate groups. Now it's 2 phosphate groups.
Right.
Okay, it was ATP, now ADP we called it. Okay. So this is the name of product. Any question?
Mr.
Yes.
Yeah, Mr. What is
You
substrate?
said that enzymes are made out of proteins and proteins are polar. Then how do enzymes like lipase digest lipids since they're non-polar?
Yeah, because here the active side, this active side has amino acids, as you know, protein made of amino acids. And you should remember that some amino acids hydrophobic and some hydrophilic, right?
Yes.
Yeah, so here the hydrophilic or let's say the hydrophobic substrates can be binded to the active site through the hydrophobic amino acids of the protein of enzyme. Okay?
Okay.
Yeah, that's it. Okay, guys, thank you for
Mr. I have a
joining
question.
today. Yeah, what?
I
Quickly,
have a
yalla.
question
You
also.
can leave, guys. Anyone want to leave is okay. Yeah. Yes.
Mr. What is substrate again? I forgot what it was again.
What is
Substrate.
the substrate? Any
Yes.
reactants need to react to each other. They bind on the active site of the enzyme. We call it substrate.
Okay.
Okay. Any two
Thank
reactants. Okay. Any two things need to react together, and they bind on the enzyme active site to do the reaction. These we call the substrate of the enzyme. The substrates of the enzymes. Okay, which they are fit to the active site of the enzyme. Ali Ruslan still here? He left, yes. Okay, that's it for today. Thank you, guys. See you later. Yeah, Abdullah Baroudi, do you have something?
Yeah, sir, can you give me like an example of enzymes?
Many examples we give, Ali, Baroudi, sorry. Enzymes like here, I gave you here examples like lactase enzyme, which is used to react the lactose with water to break it down into glucose and galactose. This is lactase. So here we have different examples like lactase, like saccharase, okay, amylase. These are different enzymes, okay. In general, next time, inshallah, we'll give you another examples. Don't worry, okay, Baroudi? Okay, yalla, enjoy your day. See you later. Bye.
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 Bye bye.